Hello, everyone. This is Juliana from National, and we are going to get started. Just so you know, we are recording this meeting. And if during the course of this meeting you have any questions, you can feel free to use the Q&A panel at the bottom of the screen. If we have time, at the end, we will answer those questions, but I'll go ahead and turn it over to Cosme. Thank you.
Welcome, everyone, and good afternoon. It is my pleasure to introduce to you Jeff Baxter, the CEO of BVI Vaccines, and Dave Anderson, the Chief Scientific Officer of the company. BVI has emerged as one of the pioneers, leading companies in the vaccine space. I will allow them to introduce themselves, but before I give a brief background on the company. They are developing a very interesting technology, breakthrough technology in the vaccine space, which is based on virus-like particles that stimulate the innate and adaptive immune responses to combat infectious diseases and cancer. It's an immunotherapy company with a very advanced product that is seeking FDA approval for vaccination against the hepatitis B virus. But today we'll be focusing on what the company is doing to fight the COVID-19 pandemic. So with this, Jeff, why don't you give a brief introduction about yourself and pass it to Dave, then we will start the Q&A session. Jeff, you're on mute.
Okay, great. Well, first of all, Cosme and Rennie, let me thank you both and the team at National for hosting us today. We're delighted to have the opportunity to talk about the work we're doing across the coronavirus spectrum. So I'm Jeff Baxter, I'm the CEO. I've been CEO at VBI for about 10 years since we were a private company. Prior to that, I was a founding managing partner in a venture capital firm for 3 years. And prior to that, I had almost 20 years at GlaxoSmithKline across a range of disciplines, including R&D, worldwide manufacturing, corporate development, business development, etc. Dave, over to you.
Great, thanks, Jeff. I'm Dave Anderson. I'm an immunologist by training. I was at Harvard Medical School focusing on cancer immunotherapy, and then left to join VBI about 6 months before Jeff did. I'm the CSO.
Excellent. So let's start by discussing in general the status of the vaccination campaign in the United States. We have 3 approved vaccines in the US right now from Pfizer, BioNTech, Moderna, and Johnson & Johnson. The Johnson & Johnson's vaccine, as we all know, was recently halted, but we believe vaccination programs with this vaccine are resuming. So in total, we're seeing in the United States right now approximately 2.7 million shots being administered daily, and 37.3% of adults in the US have been vaccinated. Later on in the call, we will address one very important subject of today, which is the challenge of emerging variants of SARS-CoV-2. But that will be addressed on the next question. For now, we wanted to ask the panelists to talk about the status of the COVID-19 vaccine field in general in the US and internationally.
Yes, so Cosme, I think well, first and foremost, for those of us that live in the United States, we should feel really lucky that the government over the last 18 months has put in place an extraordinary plan to develop, finance, and acquire vaccines to rapidly roll out vaccination programs. And I would say globally, Israel, but it's only a small country, 8 million people, are obviously the leaders in terms of creating some degree of community immunity. But the US and UK have done exceptional jobs. But tragically, as we're seeing, particularly in India and in Latin America, in those less developed companies that couldn't afford to make the financial commitment and the scientific commitment to support vaccine development. Unfortunately, the vaccine rollout is a long way behind where it should be. I think if we focus on North America, again, I would encourage everyone to get vaccinated if they haven't done so already. And what we're seeing is, in fact, data published today that of the millions that have been vaccinated, there's been about 5,800 post-vaccination infections, which is a very, very small percentage given the number that's been vaccinated, as you mentioned earlier. And I think, whilst there will be some breakthrough infection, the degree of the symptoms are lesser, clearly, after you have the vaccination. I think you mentioned the sort of vanguard group, and obviously the vanguard group have done an incredible job for society to get those vaccines out in record time. But also it's been fantastic for companies like VBI, because whereas perhaps we used to take a year in preclinical development to get a virus-like particle biologically based vaccine to clinical studies, we've actually managed to make massive process improvements such that we are now in about a 6-month development lead time. And I think that, and we're going to discuss it in much more detail later, but I think that's particularly important when you think about the variants. Now, the reason why it's so important for us to quickly get to some degree, high degree of immunity is the world is an incredibly connected place. And, South Africa strain was probably the most concerning of the more recent mutants. But more recently, of course, we've seen the double mutant, so-called India strand, emerge. And in fact, I saw just a paper this morning that outlined that some 50 people from a flight from Delhi to China of a plane of 200 people, once they got off that plane, they were COVID-19 positive. Now, it's important to be vaccinated against the original Wuhan strain, because it will prevent, perhaps not to a 95% protection level, but it will prevent transmission of the variants. And the variants and the lack of creating this community immunity will be, unfortunately, the thing that could derail this train of protecting and achieving community immunity.
Excellent. No, I saw the news on the flight from New Delhi to Hong Kong, and scientists are still scratching their heads to see how transmission could occur so quickly. And so there is a lot to learn still about the transmissibility and virulence of these viruses, especially of those new strains. Jeff, you mentioned about Israel, and that's a very important model because it's a country of only 9 million people. And from there, what we have seen is that on February, the 7-day average of infections per day was 4,117. And on April 22nd, the number of infections have dropped to 129 only. So the vaccination campaign there is working. Obviously, 50% of the population have been vaccinated. So that's the importance of raising that population immunity. So scientists are saying that if you can get anywhere from 35 to 50% of the population vaccinated, that's when you're going to start to see significant drops. Because here in the United States, we saw that plateau where cases were not really decreasing as much. The 7-day average compared to the 14-day average was not faring very well. But now this week, we are seeing the 7-day average dropping below the 14-day average. Still, the average of 50,000 cases per day was a concern. So it'd be important to know how BBI vaccines will address all of these issues, specifically how long immunity from these vaccinations and also natural immunity from patients who recover from an infection will last. The duration of that protective immunity would be very important, and Pfizer, Moderna have been talking about the design for administering booster shots. So we wanted to ask you what your thoughts were regarding the duration of immunity with these vaccines are available, and what VBI vaccines could do potentially to improve this.
Yeah, it's an excellent question, because really, unfortunately, Cosme, no one really knows at this point in time, because this has been with us such a short time. But there have been some important studies published. So, for example, when the frontline healthcare workers in the UK and the NHS were vaccinated, they followed them. And what they saw was that, and this was with the AstraZeneca Oxford vaccine, which is probably the least effective of the sort of Vanguard 5, as it were. What we saw was, the kinetics of protection, so neutralizing antibodies remained around about stable for 6 months before they started to taper off. Now, because no one knows, because it hasn't been around long enough for us to know, what the memory B cell response will be. And what that means in simple terms, and Dave will cover this as we get into more technical discussion later, but the immune system, once it's been exposed to a virus, once it's been exposed to a virus or a vaccine, it generates neutralizing antibodies to a level which may decline over time. The question is, when the body gets re-exposed to that vaccine, how quickly do these memory B cells kick in? So I've been exposed to the vaccine again, I need to kick up these neutralizing antibodies. And I don't think people understand what that kinetic is at this point in time. And so, if it's for 6 months, and certainly the Moderna and the Pfizer-BioNTech vaccines look more potent than J&J, AstraZeneca, we don't know yet about Novavax, which is last of the Vanguard 5. But before I get onto the booster question, what is really important is where you start. Because if you start, on a scale of 0 to 7, say, out of 10, then you decline from that plateau of 7 over time. And if the memory B cells do kick in, then they're kicked back up to maybe 5, 6, or 7. No one knows that dynamic yet. But if you can start at 10, you start at a higher plateau. And obviously the decay starts from a higher baseline. And that's where we believe that our EVLPs have a significant role to play in the global fight against coronaviruses, given the quite extraordinary level of neutralizing antibodies we saw in the preclinical models, some 40 times higher than some of the other published data on neutralizing antibodies. So this is not one of these questions where, biggest is best, but from what we know at this point in time, the higher your start point, the higher the plateau and the longer the decay of connection. Now, when the CEO of Pfizer said that boosters would be required within a year, I think that's absolutely predictable. All of the public health experts we talk to around the world will say, even if the decay rate is quite slow, why wouldn't you actually annually boost, particularly if you have a vaccine against the latest strain? So what we've seen is that the vaccines against Wuhan, the original strain, are highly effective, obviously, Pfizer and Moderna, 93, 95%. You don't get any better than that. But going forward, they're less effective against the variants. Now, if you develop a vaccine against the latest variant, in our case, we have a development candidate for the South Africa strain, then it's as efficacious going back to the baseline of Wuhan. And so when the Pfizer CEO or Stefan Bickel at Moderna says that a booster is required, they're not necessarily talking about a booster against the Wuhan strain, the base strain. They're likely talking about, we are developing a booster against the South Africa strain, or we're trying to synthesize the double mutant India strain, such that in a year's time we can boost. And the everyday parallel I would draw is the flu market. People particularly at risk every year get an annual flu shot, those particularly at risk. And certainly, the at-risk population should certainly get a booster. So, over 65, perhaps with respiratory or cardiovascular issues, you'd want to get them a booster. But also, and this is where, of course, wealth disparity is so heartbreaking, in the developed world or in the G7 countries, even if you're a fit, healthy 30-year-old and you had to pay for it out of pocket, it's probably worth spending $60 or $70 to get 2 vaccines just to maintain that level of protection. because obviously the societal impact of a breakthrough would be tragic. And, for the sake of those, particularly, as I say, in the G7 countries, what I think we're likely to see is recommendation from public health authorities that irrespective of whether you're at risk or not, you should get an annual booster.
That's very interesting and a well thought response, Jeff. A super interesting approach. Last year, your team at VVI showed really outstanding antibody responses from the two candidates that have tested experimentally. And some of those antibody titers of neutralizing antibodies were actually higher than what has been seen with other vaccine makers at that particular stage of development. So can you talk to us a little bit about the strengths of your technology? Why you think that this would be very efficacious against different families of coronaviruses, and also to comment on the very important positive results that you released this morning from the viral challenge study?
Yeah, I could certainly answer that with my high school science, but I'd rather hand over to Dave Anderson, who's our PhD immunologist and Chief Scientific Officer. So Dave, over to you, sir.
Thanks, Jeff. So it sounds kind of obvious to state, but when you're developing a vaccine, the better it looks like, the more faithful the mimic of the virus you're targeting, the more relevant that immunity you generate, and by extension, the more effective it's going to be. So if you think about the virus causing COVID-19, it's what's called an envelope virus, and the spike protein, which is the focus of just about everyone's vaccine candidate right now, it sits on the surface anchored in this bilayer. Well, that's precisely the way our EVLPs look structurally. And if you ask the immune system, what's the best way, the preferred way to get activated, it loves to see the spike protein or the target presented in a nice orderly array. And that's precisely the way we present it with these EVLPs. And so I think that said, it's not a surprise that if you compare the same dose of a recombinant spike protein to that presented on the surface of the EVLPs, after one dose, you have immunity, which is 3 times higher than high titer convalescent sera from people that have recovered from COVID-19. You don't see that after a single dose with a recombinant protein. You don't see that after a single dose with an mRNA vaccine candidate. So that really kind of demonstrates the potency of this way of presenting the antigens. Coming back for a moment too to your question about durability, based on this potency, we thought that there might really be a chance for a single dose, regimen, which could really help get some level of protection, as quickly as possible. Well, we asked the question, what about the durability? And so in some of the data that we just announced today, what we found is that after a single dose, we maximize these neutralizing antibody titers within 2 weeks, but they're stable with no decline for at least 3.5 months. Now, in our lifespan, 3.5 months, it's not that long a period of time. But remember, mice live about 18 months at best. And so it really is a very significant piece of new data demonstrating the durability of immunity that we can elicit with these EVLPs.
Dave, and if the data in mice, the immunity and protection lasts for more than 3 months, is it possible, theoretically, hypothetically, to speculate that in humans that duration could be much longer?
Yeah, I think it's reasonable to assume that. We're certainly encouraged, but I think as Jeff said, time will tell. I think what it does, what these data do give us is confidence that we are going to have a durable vaccine induced immunity. I think part of that stems from the adjuvant as well as the particles. We're using an adjuvant which has been used literally in tens of millions of people, including infants around the world. So tremendous safety, and it's known to induce very durable neutralizing antibody responses. And so I think that's one of the reasons we're particularly excited about this candidate that we moved into the clinic very recently.
Excellent. Let's talk now about the variants, because this is definitely a challenge and something that is a concern for for everyone. In the US, the CDC has come up with a classification of emerging and established variants of the virus. And these variants are classified into 3 groups. There is variants of interest, and those ones are determined by use of biomarkers, following outbreaks in different cluster populations and the high prevalence of these variants. Then in the second group, they identify variants of concern. And in this group, you're talking about variants that are very difficult to detect because some of the existing diagnostic tests, they don't work effectively. These variants also significantly reduce the response to vaccine and therapeutics. And they have demonstrated increased virulence from 20% to even 50%. So one example of a variant of concern would be the South African variant, B1351, that has a 50% demonstrated increase in virulence. And this variant, as Jeff mentioned, you have a potential solution or a potential product can target this variant. Of course, in this group, there is also the British variant, B117. There is the variant P1 that's being identified in Brazil and Japan initially. And there are also the Californian variants, B1, 427, 429. And then the CDC lists the third group for variants of high consequence. And this would be the worst case scenario where therapeutics and vaccine don't work at all. Fortunately for us, there is no identified variant in this group, not yet. So the question to you is, how well do you think the currently available vaccines are suited to provide protection against these variants? And how is VBI building his pipeline of coronavirus vaccine candidates? Talk a little bit about the collaboration with CEPI and NRC Canada, and how you guys are going to address this challenge.
Yeah, so I think, as Jeff just said, I think one approach is to have something like an annual boost with a new variant to ensure that you have protection. But when you think about that, you're always going to be playing catch up. And, when we first announced our program, we said from the very beginning, look, the SARS-CoV-2 virus, it's an RNA virus. It is going to mutate. We only had to look at what happened with SARS, where we knew that immunity against early strains of SARS were no longer, or were much less effective against variants that emerged 18 to 24 months later. Now, it looks like it's happened even faster in this case. So that said, we want to try to take advantage of our technology and really try to broaden immunity to anticipate, if you will, variants that were going to emerge. So whereas our monovalent candidate against the Wuhan strain, this candidate is called 2902. It's just recently gone into the clinic. We also announced at the same time a candidate called 2901. And conceptually, what we wanted to do was express not just the SARS-CoV-2 spike protein, which is the main target again, but to also express related but different spike proteins from the most significant clinically human coronaviruses. So in this candidate, we have the spike protein from SARS-CoV-2, from SARS, and from MERS. And just to help with the analogy, let's think about those spike proteins as colors. So you've got a red spike protein, a yellow spike protein, and a blue spike protein. When preclinical studies, as you would expect, you immunize this candidate into the animals, and you make antibodies against the red, yellow, and blue spike proteins. But what we really want to do, and what we've seen, is that if you present the immune system those 3 different spike proteins, you can teach the immune system to see something that's a little bit between, say, the red and yellow spike proteins. So you make antibodies that see a shade of orange, or other antibodies see something that's structurally a little bit between yellow and blue. And so now you're making antibodies that see a shade of green. In doing so, you're effectively broadening immunity. And thus far, what we've been able to see is that immunity with this 2901 candidate was able to recognize a human beta coronavirus called OC43, which is not contained within the vaccine. And so by definition, we've been able to broaden immunity. Now, when we first started these preclinical studies, it was really just the circulating Wuhan strain. And so now, as you would expect, what the team is doing, night and day, is basically getting the reagents together so that we can actually test experimentally, does sera from animals vaccinated with this pan-corona 2901 candidate actually neutralize effectively the South African variant, the Brazilian variant, the Indian double mutant variant. So that's one effort. It's something that really had CEPI very excited about this approach, because taking a step beyond just the currently circulating variants and variants that are likely to emerge in the coming months, there's a bigger question, which is, what about the next coronavirus? We've already seen 3 coronaviruses make the jump. What about the next one? Because there's certainly a half dozen at least bat and pangolin coronaviruses that haven't yet made that jump. And so, we're also looking at whether or not sera with this pan-coronavirus candidate could test and protect against some of those other coronaviruses. So that's the approach we're taking right now, and we look forward to getting that data and obviously making it available as soon as we have it handy.
Dave, that's fascinating. So from what you're telling us, we could assume that this type of approach of a pan-coronavirus clinical program could lead to products that could protect us to emerging coronaviruses, so that it could be a preventive measure to a future pandemic, let's say, and if there is another one of these class of viruses to jump in, since your product might have some cross-reactivity to these epitopes of sequences, might offer some kind of protection. Is this the right assumption to make?
That's certainly the goal, and we have some early data suggesting that there's merit to this approach, but ultimately, and to be very clear, we need to generate more data and really test experimentally. But it's something that's certainly doable, and as I said, we look forward to getting that data and advancing this candidate. We have the support financially from the Canadian government. They recognize the importance, and we will be moving this candidate into the clinic later this year.
I think, sorry to interrupt, or Dave, we can't say too much, but you might want to just talk about the work we're doing with CEPI on coronavirus X, as it were.
Yeah, so I think, what I've just kind of outlined by showing different spike proteins and trying to broaden immunity, that's one approach to a more broadly reactive coronavirus. But there are additional approaches. Instead of just focusing on the spike protein, you can incorporate additional antigens that are well conserved across coronaviruses. So it's a multivalent candidate approach. And so that's something, again, we looked at our technology, they saw the flexibility and power. We can put multiple antigens in. And so that's something that we're working on preclinically as an alternate approach to a pan-coronavirus candidate. As Jeff said, the idea would be to really test the breadth of immunity that you can elicit with these different approaches, whether it's a pan-coronavirus based on spike proteins like 2901 or a multivalent candidate. Ultimately, we wanted a broad immunity, and we're agnostic to the way in which we do so. We're just delighted that we have a technology that we think will enable us to get there.
Excellent, excellent. And is it possible to get a little bit more color from the collaborations with CEPI and NRC Canada? Are part of the same type of programs, or the different differences between the two in terms of what is being funded and how you guys are collaborating with steering committees, etc.?
Yeah, so I think, the first collaboration we announced was with the NRC when we really kind of launched the program, and it really benefited from a long-standing collaboration history with the NRC. So we worked with them on an EVLP-based Zika virus vaccine candidate. They've been incredibly supportive and, running all the preclinical studies, critically, testing using neutralization assays that are the highest quality and standard. So they've been incredibly supportive in that sense. And, beyond the NRC, as I said, the Canadian government has recognized the importance of moving candidates forward as quickly as possible. So we have their support as well. CEPI, I think, has particularly been concerned about the new variants And as Jeff said, disease X, the next one to come, a year or two from now, or whenever it is. And so they've been incredibly supportive in encouraging us to look at additional preclinical candidates, but most importantly, to get an EVLP-based candidate against this South African variant. So that's what we call 2905, and as we've publicly guided, we plan to get that into the clinic, summertime of this year. But they provide tremendous support, not just in terms of clinical development, but from a CMC aspect, it's just a fantastic partnership with them.
Excellent.
Can I just add to that, Cosme, because I think it's important for existing or potential investors and the public to understand that, the first wave of financial grants were huge numbers, right? And came out very, very quickly, particularly from Operation Warp Speed and various other things. And thank goodness the administration did that. But, this second wave of grants, and I would put both the Canadian grant to VBI and the CEPI one in this bucket, were obviously much more considered. And I think it's really important for people to understand that CEPI, the Coalition for Epidemic Preparedness Initiative, is backed by the very best scientific organizations in the world. So Gates Foundation, Wellcome Foundation, WHO, etc. And it's funded globally, from Canada, the US, and everyone else. And they did extensive diligence on our platform and our candidates. And for us, whilst we're very used to people doing extensive diligence, this was the first NGO that dug really deeply for several weeks into our technology and also VBI's people to be capable to deliver that technology. And so whilst it's a small amount of money up front, 33 million, that's a fully funded phase 1 for the South Africa strain variant. And we were the only organization that CEPI gave this grant to. So I think it's important for people to understand that. And, we're very proud of having come through that diligence and solicited that grant from CEPI. And assuming we're successful, then the next grants could be big numbers. I mean, 100 million for a phase 2, phase 3, and 100 million to support manufacturing and rapidly roll it out across the world. So this we hope is a small foundational block of a much longer term relationship, particularly with CEPI, and of course we've had a long relationship with the Canadian government. So.
That's very important, Jeff, and thank you for clarifying that. I hope that you can reach those agreements and speed up the process of development of these very important candidates. Next, we want to talk a little bit about timelines. Why you don't walk us through the timelines for each of the product candidates, and talk a little bit about the phase 1/2 study for 2902 in Canada. When can we expect to see initial data or the first set of results from that particular study?
Yeah, Cosme, great question. So, I mean, the 2902 monovalent Wuhan candidate started clinical trials several weeks ago. And fortunately for us, but unfortunately for the Canadian population, as we've read about, the Canadian vaccine rollout and procurement program has not been as effective as, say, Israel, the US, and the UK. So we enrolled this study like in 2 days. There were so many people that were so keen to get some form of vaccination, we enrolled very quickly. So we can confirm our guidance that we will be able to report, at the end of the second quarter on both the 1-shot and the 2-shot potential. Now, obviously, the study is completely blinded at this point in time, and so we don't have any results. It is in the public domain that we held our first DSMB, Drug Safety Monitoring Board meeting a week or so ago. It's on clinicaltrials.gov. There were absolutely no issues in terms of side effect. A little bit of injection site reactogenicity, but that's the same with any vaccine. So we were really, really delighted about that. So, not some of the serious side effects that you've seen with the messenger RNA and also perhaps the 2 adenovirus vaccines from J&J and from Oxford AstraZeneca. So far so good, touch wood. We have data in several weeks' time, and we look forward to sharing that. Now, 2905 with CEPI, We're in manufacturing the clinical study materials now. Assuming that goes well, then we will start that study mid-year again. It will be enrolled in Canada, and I'm anticipating it will enroll incredibly quickly. And you can assume, data 12 to 14 weeks after that. Of course, we're in the hands of third-party labs. As always, we run all of our assays in third-party labs against the most stringent assays done by third parties to make sure there's absolutely, absolute integrity around these results. So that's 2902 Wuhan, that's 2905 South Africa. Now, the pan-coronavirus candidate and perhaps the corona or disease X candidates, we would anticipate starting those in probably late Q3, Q4 of this year. It's definitely going to be a second half event. But again, you would expect data, 3 months after enrollment is complete. We're really excited about those. We can't disclose, particularly the CEPI one. As Dave said, there's some proteins that are common across all coronaviruses. So if you can address those proteins, then maybe there's a holy grail across not only the variants and the 2 pre-existing coronaviruses that have been human transmittable, but against the next one, wherever that may come from. I mean, there's medical bodies around the world that are flying drones into bat caves and in jungles collecting all kinds of samples, just with the WHO trying to estimate. And obviously all of that work and that data gathering goes into our development thinking.
That is very interesting, completely brand new information. So it's good to see that the company is making a speedy progress in all these programs. Of course, situation, we hear in the news the situation in Canada with Ontario in full lockdown, how the vaccination campaign has not gone well there, but I guess the CEPI and NRC are fully aware of these, and that's going to help BBI vaccines make progress with all of these programs. So the next question that we wanted to ask you, Jeff and Dave, is regarding design of some of these trials. And the discussions with regulators, especially given that emergency use authorization in recent months have been given after relatively large clinical trials in the tens of thousands of patients. So based off what you have learned from your discussions with the team and the agency, is there any way to streamline and expedite these programs to get through the clinics faster?
Yeah, it's a great question. So first and foremost, these phase 1, phase 2 studies, we are still able to use placebo, as no standard of care per se has been established. Although for the phase 2B study, likely we will go head-to-head with one of the current vaccines, whether it be Moderna or J&J or whatever. But phase 3 is obviously the studies you referred to that consume a huge amount of time and a huge amount of money. We've actually seen between the Vanguard 5 now, with some of them having between 30 and 60,000 people in their phase 3 studies. There's now over 200,000 people have been in a controlled study environment across those 5 candidates. Now, 200,000 people sounds a lot of money. Sorry, it is a lot of money, but sounds a lot of people. But you need time to establish a correlate of protection. And the reason that this is important is that you can move from a prevention of disease and symptoms type of study to an immunological correlate of protection, where if you take someone who's seronegative, they don't have any neutralizing antibodies, you vaccinate them, they get above the hurdle of protection. And then you can run studies for 3,000 to 4,000 people. Now, you would think that with 200,000 people, you could establish a correlate of protection. The key thing is actually how many people get infected after vaccination, so that you can look at their neutralizing antibodies and establish how the virus was able to break through. And as I mentioned earlier, the rate of infection post either first infection, natural exposure, or vaccination is still low. But we are getting there, Cosme. In fact, it's a very timely question. It's in the public domain that the US Pharmaceutical and Biological Trade Association, BIO, is actually having a meeting with the NIH on Monday, just in a few days' time, to really push this and say, can we have a timetable? Because for companies like us and for CEPI and the WHO, you know, the FDA and everyone else, what they want to do is get rapid trials in place with these variants so that you don't have to enroll 30 to 50,000 people. And, you know, correlates of protection against many other common and contagious viruses, such as, for example, our hepatitis B program. We enrolled 4,000 people, you know, neutralizing antibodies. You establish what the correlate of protection is, which has been established for decades now in hepatitis B, and just prove that you can increase people's immunity above that hurdle of protection. Now, there is just one other thing that I'd say, which of course would be a huge step forward and very brave for the world to take. But of course, the quickest way would be a human challenge study. And a human challenge study is actually recently just kicked off in the United Kingdom. And I think that is really, really interesting because then you're talking about hundreds of people who actually get infected, or at least are exposed to a virus or a variant in a challenge chamber. And then they actually, very brave of these people, A, to get exposed, but also then they sit in a hospital for 6 weeks and you look for when not any symptoms break out. But there you're talking about hundreds of people and you're talking about 6 weeks rather than even 2 to 3 months for a 3,000 to 4,000 study. So I think that is really interesting. And, you know, everyone is watching what's happening in the UK in this first human challenge study.
Excellent. So given all that we have discussed today, Jeff and Dave, from the global vaccine rollouts, the pipeline of coronavirus vaccine candidates, where do you see VVI fitting into the commercial landscape, assuming that you get approval in the US and other jurisdictions internationally? What be the label of the product, or let's say, what is the targeted population and how will you fit in the competitive landscape overall?
Yes, so starting off with 2902, the monovalent Wuhan strain, our dream target product portfolio is a single dose at 5 to 10 micrograms, which is very well tolerated, that doesn't have, you know, the post-messenger RNA type of adverse events that many of us have suffered and been through. You know, convincing the world to get vaccinated is really important, and the horror stories, you know, post-second vaccination that some people, you know, are suffering will only, you know, increase that vaccine hesitancy. So I think, you know, safe, well tolerated, without the shivers, fevers, and everything else, at a very low dose that can be distributed according to normal vaccine refrigerated kind of storage requirements is our dream target product portfolio. Now, of course, 2905, the South Africa strain, is really important. And the reason is because, as I mentioned earlier, if you can protect against the latest variant, chronologically going back, you'll protect all the way back to the baseline. 2902 can have a really important role post the Vanguard 5 in markets where the Vanguard 5 haven't really started distributing the product, or you don't have deep cold store requirements. But I think the really promising candidate in the US and in the sort of 7 major markets around the world will be 2905, and then the pan-coronavirus or disease X. And I think the reason that's so important is that, you know, the world needs to get back to some degree of mobility and societal norms. I don't know what that's going to look like in the future, but it needs to get back to some degree of mobility. And so these viruses, they don't know any political or geographical boundaries. It will spread. So let's get everyone protected against the Wuhan strain. Let's get the South Africa strain vaccine out there quickly, and perhaps commercially, whilst 2902 is going to be very important outside of the US and probably outside of the 7 major markets. But 2905, I think, could be the booster of choice. Bear in mind that, Moderna are administering 100 micrograms of vaccine in 2 shots. If we can administer just 5 micrograms in 1 shot and achieve higher neutralizing antibodies without all of the side effects, without the supply chain issues, then that would be a massive breakthrough. And I think that will be a very important product in the US and in the 7 major markets.
That's excellent. Jeff, there is a question that came from the line from an institutional investor regarding the changing in the landscape in the US. It is known from scientific hypotheses that between 35 and 50% of a fully vaccinated population, you start to see a significant decrease in the number of new cases, transmissibility, etc. So assuming that how dynamic the COVID-19 landscape is, by the time that you run into the late stage phase 3 clinical trials, it will not be the same situation in terms of emergency use that the US was facing when Pfizer, BioNTech, and Moderna went in front of those FDA panels back in November and December of last year. So the question came, how do you see running those trials if the number of cases in the United States are significantly declining? Obviously, it would be more difficult to measure efficacy. So give us a little bit of color what a phase 3 clinical trial will look into the future when you get there.
Yeah, the question, provides an important insight, which I should have explained was behind my last answer about how I see this commercially. Because 2902, the Wuhan strain, I agree with behind the concept of the question. You cannot, it will be difficult to enroll a study in the US. But, in Brazil, Mexico, Latin America, Southeast Asia, India, it's going to be very easy to run studies in naive populations for 2902. Now, 2905, the important thing here is that you can specifically identify neutralizing antibodies against the South Africa strain. And this is where the correlate of protection is really important. So rather than prevention of infection, because it would depend upon the underlying rate of South Africa strain circulating in the US, then you would look for specific neutralizing antibody responses against that South Africa strain. And assuming you had identified a correlate of protection, you exceed that, you can get essentially approval on prevention of infection rather than prevention of disease. And as I said, there you'd be looking at 3,000 to 4,000 people in numbers. But certainly, if you look at many parts of the world, tragically, there's plenty of opportunity to run significant studies in naive populations. And of course, this is where CEPI and the WHO with their global reach is going to be really, really important for us to rapidly identify partners, whether it's Butantan in Brazil, the equivalent in Mexico or Burmex or whomever. But, this is where CEPI and WHO will play a really important role. But commercially in the 7 major markets, I go back to what I said earlier, 2902, the Wuhan vaccine, is not likely to be relevant, but 2905, the South Africa strain, will definitely be relevant in those 7 major markets, high price markets. But of course, with a relatively low cost of goods, particularly if you tech transfer to a local partner in Latin America or India or whatever, there's still, the numbers are huge, Cosme. So there's still a very interesting potential return to be made for the company on the 2902 Wuhan vaccine.
Yeah, no, it is understandable. The situation in India has surpassed Brazil and United States, and it's become a real tremendous healthcare crisis. And now the Brazilian strain, the P1, and there is another one, the P2, is ravaging Uruguay and some of the other South American countries. So not the same situation internationally with what you see in the US, Israel, and the United Kingdom. Even some of the other European countries are struggling to contain this pandemic. And of course, there is always the significant challenge that we might see other emerging variants with specific mutations in the spike protein that affect interaction with the receptor, increased transmissibility, virulence, severe disease, and these things. So it's difficult to predict how this thing is going to evolve. So we're very encouraged by the progress you've made and the very important products that you are developing. We thank you, Dave and Jeff, for very insightful discussion. At this stage, I'm going to pass it on to Juliana to see if we have more questions from the line. Juliana?
Yeah, thank you so much, everyone. This has been really, really great. We do have quite a few questions on the line, so it might be best, Cosme, if you picked a couple so we can, stay cognizant of everyone's time. Unfortunately, we will not be able to answer every question, but we really, really appreciate it. So yeah, if you could pick just like a couple of questions, Cosme, that would be awesome.
Thanks, Juliana. Unfortunately, I'm not getting the same feed as you, so I'm not seeing the questions here on my system directly. So only the ones that came to my email.
Oh, okay. Well, Jeff and Dave, are you seeing these questions?
Yeah, I'm seeing these questions on the Q&A. There's a couple here that I can certainly answer very quickly. There was a question, could and should VBI seek funding from the US government? That is an excellent question. Obviously, all of the effort, which was a fantastic effort by the US government, behind Operation Warp Speed, focused on these Vanguard 5, and thankfully for the population in the United States, they did. But there's a second and third waves, and we certainly will be seeking funding from the US government. There's a little bit here around, we have to be careful from a governance point of view that we don't get accused of double dipping, because, our programs have been so well funded by the Canadian government and by CEPI at this point in time. But of course, if we were to move forward with a coronavirus disease X type of program, I'm absolutely certain that that's something that, DARPA and BARDA, the US military type of groups, and the NIH would be very interested. Because when you think about this, if you could come up with a disease X, then in terms of stockpiling for military and various other applications, it would be a fantastic solution to those people who maybe have to quickly get on a plane and go somewhere. So, this disease X thing, for the sake of humanity, but, priority obviously would be healthcare workers, military, and obviously a huge private market for people that want to get back to recreational travel or for business travel. There's a question here around what kind of cell line does VBI use. We use a HEC 293 cell line that we originally in-licensed from the NRC. And it says, does VBI is manufacturing adhere to the Vatican guidance of the avoidance of fetal cell lines? It's a very good question. And we actually do have a certificate from the Vatican allowing us to use this particular HEC-293 cell line, which was, as I say, originally in license from the NRC.
Jeff, there's a question there about thermal stability, and I think
Sure, Dave, over to you.
Yeah, we alluded to it earlier. I mean, as we know, for these new classes of mRNA-based vaccines, they have to be frozen, sometimes at ultra cold temperatures, -80°C, which is just not standard. And to be quite frank, it's a real challenge and strain for more rural locations. So based on an early clinical candidate that went into the clinic using the same platform technology, we know that our candidates should be stable for 3 or 4 years using just standard refrigeration, which is standard around the world. So we think much better thermal stability with this candidate relative to the mRNA-based modalities, at least.
Dave, there was a question also about T-cell immunity, which I know you've done a lot of work on.
Yeah, so I think if you look at the publication should become publicly available any hour or day now. It's already been submitted. We did measure T-cell responses, the kind of Th1 type response. We do elicit T-cell responses. It's really difficult to quantify and to say this is the threshold that really means something. In other words, there isn't any known benchmark for how good is the T-cell response. You just want to have them because they are important to have durable, long-lasting neutralizing antibody titers. So we've measured them, we see them, but I think more importantly, as I said, there's clear data showing neutralizing antibody titers, they peak within 2 weeks after a single dose, and they're very well preserved for as long as we've followed them so far.
Excellent. Is there any other questions, Jeff, that you saw there that we have time to discuss? We still have a couple of minutes left.
Sure. There's a question here about can VBI expect a second round of funding from CEPI in the coming weeks? I think, as I mentioned earlier, I think there's potentially hundreds of millions on offer from CEPI. They're extremely well funded from all nations around the world, WHO, Gates, etc., etc. I think, we have to walk before we can run. And I think the next time that, assuming we're successful, the next time that we would actually hear about a next round of funding would likely be kind of the end of Q3, early Q4. Perhaps in the last minute, and to end this on a slightly humorous note, there's a question saying, where can we buy the BVI vests that we've seen presenters wear? Unfortunately, you can't buy those vests. We had them made as a year-end gift for all of our staff who have just been working unbelievably in a flexible and dedicated way, 2-3 shifts a day, 7 days a week to get us where we are. So it was a little year-end reward for everyone who'd been working so positively as part of this issue. So, Cosme, just one other point I just want to leave people with, and a bit broader, people have dialed into this because they're interested in the general coronavirus sort of environment. When you look at what's happening in India or in Latin America at the moment, people should never think this is just an Indian problem or Latin American problem. It is a global humanitarian crisis, and also has a massive, massive economic impact. So if you think about, the amount of global industries that are supported just out of Bangalore, for example, in India, where these populations of very intelligent, highly economically productive workers are not able to work, we're seeing all kinds of impacts, and including impacts in production of raw materials that we need every day in our labs from Latin America or from India or wherever to conduct our preclinical studies. So I think, we all, in our hearts hope that the world makes rapid progress, and 2902, our monovalent Wuhan, I think could be a really important asset to those countries, given its near thermostability, light refrigeration, a very low dose without any kind of side effects at a single shot. And I do hope that in a year's time that people are able to have a booster in North America and Northern Europe, which is 2905, the South African variant. So I think with that, it's dead on 2:30. Dave and I want to thank you, Cosme, thank Rennie, thank all of the people at National for hosting us today. And we've really enjoyed it. We're very proud of what we're doing and we enjoy talking about it.
Jeff, thank you very much, and Dave as well. It was a very lively and insightful discussion on a very important, relevant, significant topic. So we want to thank all the organizers, and with these, we conclude our meeting. But thanks, very inspiring, and we wish you all the best to the entire organization. Hopefully you can get all the funding required to finish these programs as quickly as possible with the best possible clinical results to save lives, which is at the end what we are all here for.
Great. Thanks very much, Cosme.
Thank you very much.
Thanks, team.
Thank you.
Bye-bye.